This is Dr. Jessica Han. In this mini lecture, we are going to talk about non-Mendelian genetic types. Before we go to that topic, let's review the general genetic laws from Mendel. Mendel discovered three fundamental principles for genetics: law of dominance, law of independent assortment, and the law of segregation. The law of dominance states that when two different alleles are present, one may mask the expression of the other. The dominant allele is expressed, while the recessive allele is not. If uppercase A is dominant and the lowercase a is recessive, then the heterozygous combination of one uppercase A with one lowercase a will show the trait associated with uppercase A. As shown in the image, uppercase A codes for the red color of the fruit. The lowercase a codes for the yellow color of the fruit. The heterozygous combination of one uppercase with one lowercase a will show the phenotype of red color, which is the same as the uppercase dominant allele's trait. The law of segregation states that each organism carries two alleles for each trait, one from each parent. These alleles separate during gamete formation, so each gamete carries only one allele. The figure here below shows an example of law of segregation. There are two heterozygous individuals are married. They are both carry a disease gene, which is the recessive gene little r. They are both carriers because they look normal. Even though they carry the recessive gene, the trait of the recessive gene is being covered by the normal gene, big R. When these two people get married, the gamete combination will show as below. We'll have one homozygous big RR, which is unaffected person. Then we have two heterozygous big R and the little r. They are still carrier because the recessive gene is being covered by the dominant gene. The homozygous of two recessive genes, little r and little r, will result in a diseased individual. In this case, the alleles in each individual, the big R and the little r being separated, then they randomly combined with each other to result into these four different genotypes. This example demonstrates the law of segregation. The third law is the law of independent assortment. This law states that genes do not influence each other with regard to the sorting of alleles into gametes, as long as the genes are on different chromosomes or far apart on the same chromosomes. And this physical basis for the law of independent assortment also lies in meiosis 1, in which the different homologous pairs lined up in a random orientations, then being separated. As shown in this figure here, we have homologous pairs of chromosomes are lined up in the equator of the cell. Then the chromosomes are being separated. The blue goes to one cell. The other chromosome of the homologous pairs goes to the other cell. The other alternative for separation is one blue, one red can go to one cell, and the other pair goes to the other cell. So the figure demonstrates the random and independent assortment of those homologous pairs. Now let's look at the examples of non-Mendel's genetic types. The first genetic type that does not follow Mendel's genetic laws is incomplete dominance. Incomplete dominance is the situation that one allele is not completely dominant over another allele. For example, if big R coded for red color of the flower and the little r coded for white color of the flower, the heterozygous RR will result in a blended color, like a pink color. And here is a picture of the incomplete dominance. So the homozygous red flowers are coded by RR. The homozygous little r result in white color of the flowers. The gamete for each type of flowers are being separated, then they randomly combine to each other, then the phenotypes for their offsprings are pink flowers. This situation is called incomplete dominance. The second type of non-Mendel's genetic case is codominance. Codominance means that more than one allele can be dominant and expressed. The best example is the blood type. Alleles coded for A and B can be expressed at the same time. That's why we have a blood type AB. Inheritance of the ABO blood system in humans is complicated in that multiple alleles are possible. So we normally express the dominant allele, use the uppercase I. Then we use the lowercase little I to express the recessive allele. Homozygous of 2 little I will result in a blood type O. In this figure, it shows the combination of different alleles. When we have 2 alleles which are dominant for A, we'll have blood type A. If we have dominant A with dominant B, we'll have blood type AB. We have A with little I, we still have blood type A. Blood type A can be the result of 2 genotypes. One is homozygous dominant IAIA, big A, or they can be heterozygous big A and little recessive gene. The same thing for blood type B. When it comes to the blood type O, we'll have 2 little i's. For blood type A, on its red blood cells, they have the A antigen. In the plasma of the blood type A, they have the anti-B antibodies. In blood type B, it has B antigen on the surface of the red blood cells, and in the plasma, they have anti-A antibodies. And for blood type AB, it has both antigen A and antigen B, and there's no antibodies in the plasma. For blood type O, the red blood cells do not have any antigen, but in their plasma, they have anti-A and anti-B antibodies. Since the blood type O has antibodies to against A and B, he can only receive the blood type O as a blood donor. For blood type AB, since there is no antibodies in the plasma, he can receive the blood from blood type A, blood type B, and the blood type O. The third non-Mendel's genetic type is an X-linked traits. In this case, the traits that controlled by genes are located on the X chromosomes, and the X-linked traits always follow a unique inheritance patterns. The inheritance of this type of trait located on the X chromosomes often goes with the sex of the individual. For example, the red-eye fruit fly is crossover with a white-eye homozygous fruit fly. Big W coded for red eye and little w coded for white eye. For the female, she has 2 gametes, X little w, X little w. For the male, he will have X big W, which is dominant allele, and Y. Then the combination of those alleles will result into 4 different types of genotypes. We'll have X big W, X little w. And this female still have red eye, which comes from the father. And this female, the same thing. For the male fruit fly, the X little WY will result in a white eye. The same result for the other male. So all the male offspring will have white eye because they carry the X little w. The next type of non-Mendel's genetic situation is linked genes. Many alleles are located in the same chromosomes. While the homologous pairs being separated, genes on the same chromosomes will be linked together to be passed into the daughter cell. As the distance between 2 genes increases, the probability of 1 or more crossovers between the homologous pairs can increase, and the genes behave more like they are on a separate chromosomes. So crossing over will create genetic variation. Here's an example to show you how the alleles on the linked gene being separated. If the genes are on different chromosomes, based on the independent assortment law, there are 4 possibilities for gametes. So you will have big AB together, little ab, little a, big A, little b. Will have equal percentage for each of the gamete. However, if A and B are in the same chromosomes, the separation will be different. The first possibility, the big A, big B will be together. The second, A and B will be together. If there's any crossing over happen, then you will have a possibility of big A, little b, and little a, and big B. But the crossing over depends on the distance between these 2 alleles. If the distance is large enough, the last 2 gametes are possibly to be made. However, if A and B are very close to each other, there will be only 2 gametes produced, which will be the first 2. Then the crossing over will have a very rare chance to occur. Another type of genetic situation that does not follow Mendel's law is epistasis. Many traits are going to be controlled by the expression of multiple genes. For example, the eye color in humans is controlled by at least 8 genes. In epistasis, the interaction between genes is antagonistic. One gene masks or interferes with the expression of another gene. For example, the white type coat color big A, which shows in the brown, is dominant to the solid black color, which is little a. However, a separate gene C, when present as the recessive homozygote little c, it will negate any expression of the pigment from the A gene and result in a white albino mice. In this situation, the C gene is epistatic to the A gene. This table summarizes all these various types of non-Mendelian inheritances. Incomplete dominance, codominance, multiple alleles. We mentioned that in the previous lectures. Polygenic inheritance. This is the situation that traits controlled by multiple genes. For example, the skin color of humans, the height of the humans. Pleiotrophy. This is the situation when one gene affects multiple traits. We already talked about the epistasis using the coat color in mice and X-linked problem. Mitochondrial inheritance is a situation that genes are inherited only from the mother, well, the mitochondria. The genomic imprinting states that expression of the gene depends on whether the gene is inherited from the mother or the father. Anticipation is a situation that genetic disorders get worse or appear earlier in successive generations, such as Huntington disease. This table will give you a good summary for the types of genetic situations which does not follow the Mendelian law.